Okay, continuing with cardiac muscle. So the cardiac muscle contracts. Now, again, one of the differences between skeletal muscle and cardiac muscle is that skeletal muscle needs a nerve impulse to start the contraction, to set off the action potential and result in a contraction. To kind of review for just a second, what happens is in skeletal muscle, the motor neuron releases acetylcholine into the cleft, into the synaptic cleft. The acetylcholine binds to chemically gated, ligand-gated channels, sodium channels, on the sarcolemma. They open up to sodium. When the sodium enters the cell, it depolarizes the sarcolemma, which then causes the voltage-gated channels to open in a wave of depolarization called an action potential. That moves down the T-tubules. The depolarization of the T-tubules depolarizes the sarcoplasmic reticulum, which causes the release of calcium. The calcium binds onto the troponin. The troponin pulls the tropomyosin out of the way, exposing the binding site. Myosin binds to actin, power stroke, contraction. Cardiac muscle does much the same thing, except that instead of ligand-gated channels, some of the channels in some of the cells, in about 1% of the cells, there's actually leakage channels. There are channels for sodium that are always open. The sodium-potassium pumps pump the sodium out, reset the resting membrane potential, and then sodium leaks back in, and when enough leaks back in that it reaches a threshold, it depolarizes, causes the voltage-gated channels to open in a wave of depolarization that's an action potential. And from there on, it happens the same way. So the thing is that these are automatic. You pump the sodium out, it leaks back in, reaches the threshold, action potential. Pump it back out, causing relaxation, it leaks back in, reaches the threshold, action potential. So it's rhythmic. The cells are self-excitable. And because of the gap junctions, the action potentials in those 1% of cells pass through the gap junctions and cause depolarization and action potentials in all the other cells, which means that the heart contracts as a unit. Now, if you remember in cardiac muscle, the refractory period was very short. The absolute refractory period was quite short because there was a lot of sodium-potassium pumps that reset the resting membrane potential. In cardiac muscle cells, there are fewer sodium-potassium pumps, so the absolute refractory period is very long. It's actually a quarter of a second, 250 milliseconds, which is a very long time. So the heart has a lot of time to have its action potential, have the contraction, and completely relax before it can have another action potential, before it can happen again. And this ensures that the heart completely relaxes and can fill with blood before the next beat. You can't have tetanus. In skeletal muscle, tetanus is important because it maintains muscular contraction, so we can carry things, we can maintain our posture, etc. If you had tetanus in your heart, there would be no pumping action. You need to have that relaxation time to fill, that quarter of a second to fill, and so that's why we have this long refractory period. If I if you don't think that I've emphasized that enough, think again. The long refractory period is vital to the proper functioning of the heart. What it means is the heart can't pump any faster than 4 times a second, which at 60 seconds to the minute means that we have what, 4 or 300 and whatever, 122, 240 beats per minute is the fastest that the heart can actually beat. Okay, so depolarization gets the voltage-gated channels open. You have it's exactly the same. When the one of the things that happens is the sarcoplasmic reticulum releases calcium, but there are also calcium channels open in the sarcolemma. So calcium that's outside of the heart in the pericardial space can also come in. So that depolarization phase is prolonged because there's more calcium available. So it contracts and stays contracted for a second, and then it takes a little bit longer to reset. So the exact same thing. Calcium binds to troponin and pulls the tropomyosin out of the way, and you end up with contraction. So the duration of the action potential is longer than in skeletal muscle, and the contractile part is therefore longer. And then we repolarize. Now, to coordinate this, there are special cells within the heart that are muscle cells that act pretty much like nerves. They are elongated cardiac muscle cells that have few, if any, myofibrils, and their job is just to transmit action potentials via gap junctions to the other parts of the heart. So they coordinate the beating of the heart. They coordinate the timing of it. And this is called the intrinsic cardiac conduction system. Now, what's interesting is that because of this autorhythmic cells and this intrinsic cardiac conduction system, that means that the heart will beat even if it's not hooked up to the brain. This is why we can actually transplant hearts, because the heart will beat independently. You can pull the heart out of somebody and put it in a petri dish, and as long as there's oxygen and nutrients, there's some sugars, it will beat on its own. Every Aztec priest knows that. The heart will beat outside of the body, and therefore could be transplanted. And that's really because of this cardiac intrinsic conduction system. So these are the non-contractile and they're autorhythmic cells, and they coordinate the contraction of the heart. I've already talked about this. So they autorhythmic have unstable resting potentials, so they have open sodium channels. We call these cells that have this pacemaker cells. So it follows this, and it's rhythmic. So the first place we have a bunch of these cells, pacemaker cells, gathered together right on the wall of the atrium, of the right atrium, right beside where the atrial sinus comes in. So we call this the sinoatrial node, the SA node. And it generates impulses about 75 times per minute. It's the fastest autorhythmic depolarizer, so it sets the pace. So left on its own, the heart will beat 75 times a minute because of the SA node. when the SA node, when these cells fire, they set off an action potential that spreads through the myocardium of the atrium, and it gets down to this place called the atrioventricular node. Now, there are no gap junctions between the cells of the myocardium of the atria and the myocardium of the ventricles. The only place where this action potential can transmit is through the AV node. There's smaller fibers, there's fewer gap junctions, and what happens is the action potential gets delayed there for about 0.1 seconds. Now, if the SA node is not functioning, then the AV node can take over. But unfortunately, its rate of depolarization is 50 times per minute. So it's kind of the backup, but it doesn't normally set the pace. It only sets the pace if the cells of the SA node are non-functional. Its main job in a healthy heart is to delay the impulse and to be the only connection between the atrial myocardium and the ventricular myocardium. Now, the AV node attaches to a whole bundle of elongated cells called the AV bundle, or the bundle of His. I don't know what His stands for. You will see it a lot. I, as a student, in my mind, just to remember it, thought of it histologically in the interventricular septum, HIS. It's the electrical connection between the atrium and the ventricle. It almost acts like a bundle of neurons to be like a nerve, except they're not neurons. They are cardiac muscle fibers, just non-contractile. Now, as the AV bundle goes kind of into that AV septum, or through the AV septum and into the interventricular septum, it branches into right and left bundle branches. One goes to the right side and the other one goes to the left side, obviously. They both carry on down the interventricular septum to the apex. At the apex, they turn up the myocardium as Purkinje fibers. And the Purkinje fibers move along the walls on the outside of the ventricles. And now they depolarize at 30 times per minute. So if there is no AV input or if one of the bundle branches is blocked, they will still depolarize, but only about 30 times per minute. So it looks like this. We have the SA node spontaneously depolarizes. It spreads through the gap junctions through all the atrium, causing the atrium to contract. The impulse is delayed here at the AV node for about 0.1 seconds, and then starts to transmit down the bundle of His and through the bundle branches. And you'll notice that things go up here into the papillary muscles. They go to the apex and then along the myocardium this way as Purkinje fibers. This delay allows the atrium to contract and actually start and push blood into the last bit of blood into the ventricle, and then start to relax before the ventricle starts to contract. And you'll also notice that the first part of the ventricle to contract is going to be the septum, because it's going to get the impulses first. So that's actually going to pull the apex up towards the valves and start pushing the blood this way before it starts squeezing from the sides that way. And it ends up squeezing the blood here. And you'll notice that the semilunar valves are kind of to the medial side. So this squeezing from the outside pushes blood directly towards the semilunar valves. So the heart will beat on its own because of this intrinsic conduction system. But obviously, your heart rate changes. When you're exercising, it goes up. When you're at rest, it goes down. If you're in good shape, it might be beating 60 times a minute. It might be even less. It might be in the 50s. If you're excited, it might get into the hundreds. So the heartbeat is modified by the autonomic nervous system. And it's controlled from the medulla oblongata. And there's cardioacceleratory centers and cardioinhibitory centers. So the acceleratory speed up the heart rate, and the inhibitory slows down. And really what it does is it innervates the SA node and the AV node, the acceleratory through sympathetic neurons, and the inhibitory through parasympathetic neurons, the vagus nerve. So what happens here is that you have the cardioinhibitory, which comes out through the vagus nerve, and then goes to the SA and AV node, and actually causes the closing of some of those sodium channels, so it takes longer to reach depolarization, and the heart rate slows down. Sympathetically, it goes through the sympathetics and then opens channels that makes it easier, and you reach the threshold sooner, and the heart rate speeds up. And normally the heart is beating at a rate that is the balance between these two things. Normal, relaxed, the heart is more under vagal, under the parasympathetic, and it slows down. So we say the heart has vagal tone. The heart rate is usually less than what it would be without the inhibitory effects. I have already recorded the ECG, so we're going to stop this one here.